What is the group? As people have found the group, then we take out a sheet of paper and we can circulate, and then we can put the names in the group. I mean, the groups that they are interested in. So just pass on one sheet. Put like group A, group B, like this, and leave a blank for, yeah, you can put like this, and then a half page. You can put in group A, group B, group C, group D. We will have a good work. It's up to us how we want to do it, right? And this is really incredible, as we discussed last time, that involving ourselves in a research is the best thing you can do in gaining skills of different types quickly. Okay. So what he's doing is he's circulating the sheet of paper. Why don't you guys, are you guys from the same school or different school? Same school. So put yourself name in a different group, not in the same group. Okay. Yes.
Sir, I have researched around 13-14 topics.
Okay.
I will give you share on the WhatsApp group if someone.
Okay. Why don't you?
Rough guide I have made because of lack of the time in the morning.
Fantastic. So this is exactly what I wanted all of you to think about. See, I know he has the experience. Thank you very much. I know he has a lot of experience in the. But when someone is, this is a course to learn, right? To do the research work and to gain skills, you have to be proactive. You know what is proactive? You have to be active in advance. If I say one word, you know how to complete the full sentence. This is how the sharpness in the research comes into picture, and in the job market as well. Okay, so what I expect, so he has done exactly what I was expecting. So what he has done is, he has not only thought about who could be his group members, but also, again, that's a different issue whether I let him do his own group or not. That's a different issue, right? I want to be very diverse group, each one of us, right? In a very diverse group.
Can I share those?
Right. So I'm coming to that. So, and then he has also done the second part, which was thought about potential research things that he can work on. Okay. So, okay, go ahead and talk about top 4 of them, or top 3 of them.
So, how to present?
Just talk in couple of sentences. What are you thinking about doing as a group project, and if there are any takers. So you are presenting it in a way in 2 or 3 sentences or 4 sentences at the most, so that others can get attracted to work with you in your group also. Okay. So go ahead.
So firstly, there was one topic. It's based on the youth culture that is in India. It's on the social experiment. Okay. So it is like that. There is one sibling. They have made a band, but that band doesn't perform rock concerts or like Bollywood or top songs. They perform bhajans. So mostly some of the Gen Z youth, which we say in today's world, they are getting attracted to that bhajans. They are booking tickets and all. And one part of the Gen Z youths are following social influencers and all. So why is this distraction happening? Why are frame shift happening? Then the second topic is basically mostly now, you must have heard now these days it is in news, there Aravali Hills mining. So government is taking money from the big investors allowing mining, but the people there in Rajasthan, they are protesting against this. So it can be another topic.
But what is the challenge that you are?
Challenge is that if that hills.
The first challenge was clear. This one I'm not.
If the hills are cut down, then environmentalists predicted that Delhi can turn into a cold desert. And the second point in this is that the AQI levels in Delhi and around NCR regions will, if that's the cause affecting it can be another reason. And the third reason was about the people of that area, their livelihood and all.
Okay.
Environmental concerns.
Okay, so these are good topics to model. Though, again, actually I'm very impressed with the amount of work that you have done and brought some different topics. And these are practical challenge. I really like the topics that he is latest cutting edge is impact all of you, even if you live in a different part of the world and not in Rajasthan where Aravalli hills are there, or he's talking about Delhi where AQI is very high or something like that. But still, it is a relevant topic of today's affairs, right? So what we could do here is, one thing to remember when you are choosing a topic. I talked about yesterday an example of social problems, right? Social unrest, such as Gen Z, this thing, riots in Nepal and Bangladesh, for example. Now, why did I give this example when the course title is on infectious disease and global health? Why is it a really relevant issue? What is the connection with the two? Remember, I'm not going outside the arena of the course topic and title, and I'm still fine with the type topics that he has talked about. First of all, health comes in a very big ways. Health doesn't mean only from diseases. It comes from environment. It comes from other chemicals, right? So he's talking about relevant topics that is really important from the health perspective. That is one point. But that's not the only point. What was your first topic?
There is one siblings. I don't remember the Instagram of them. They are into bhajans. They are making rock band of bhajans. So mostly some of the Gen Z youths are getting attracted towards it, and the other ones they are following big influencers.
Okay.
So what is the distraction? Why is this happening?
So this topic, you all heard it, right? How is it related to diseases that we are modeling?
It's influencing the mind.
Does it make sense what he's talking about? How is infectious disease spread? How is infectious disease spread? By infection. And infection is spread by one coming in contact with other, coming close contact with other, right? That's how infection is spread. What he's talking about is spread of ideas, information, which is similar, exactly the same way. Does that make sense? So that's why this topic is very relevant, because it is also an infection of social ideas and social views. And that's why yesterday's example that I gave on Gen Z was also relevant, because why did I say about tipping points? The group comes into picture in certain ways that tips off the system. Does that make sense? So social issues are also very much relevant to this fact. Okay. So what I will do is, there is a sheet somewhere. Where is the sheet? Okay. Have you guys written the names? Okay. So also, all of you are from maths department and some anthropology and some biomedical. Anyone else from a non-math background? All of you are from so mathematics, except these two folks. Okay, that's fine. So the thing I'm thinking about is, once you will write it down, your names in different groups, I will going to ask your skill set. I don't want same skill set people in the same group. I want different skill set people so that you can complement each other. Okay. So anyway, I'm circulating this sheet is, you write down your names and pass it back, and do the same thing. Try from same institution not to be on the same group. Try. Okay, if it, I mean, we can't guarantee everyone to be, to cover in that 6th group. There are 6 groups, so we have now, how many folks total are there? Do you guys know? Where is 45 physically are here, right? 45. So 45, and so roughly around 7 folks per group will be there, which is decent. So there is 6 group we are aiming for, so 7 folks. So Have a seat, have a seat.
The third topic.
Oh, you have a third topic.
I have 13, I have done for 13, but now it's on the drug.
But remember, these two are the focus. Health and infection process. That is the two goal of you to choose the project objective. Now, so today the goal is we will give you some time in the group projects, and you guys will discuss and think about one of the projects that research objective. Not only topic, but also topic. Forming a group was yesterday's task as a homework, and today's task will be the research objective. So choosing a topic title. Okay. So you will discuss among the group members and see if you can come out in the evening. We will do it, right? And you will have more idea because I have some example in my slides, and you will see more. Is that okay? Okay.
So the third topic is BHU researchers have found out a plant in the Himalayan region. It's in Hindi it's called Patthar Chitta. So it has an antioxidant which is named as rutin. So they are developing a drug using that antioxidant for the treatment of cancer.
Okay.
Then the last topic is my brother-in-law. He is a doctor in AIIMS Delhi. So he is working in the department of microbiology. So they are working on antimicrobial resistance. Like people today, they take, when they have cold or fever, they take medicine roughly without prescription of doctors. So they are making some research like good bacteria, bad bacteria.
Okay.
So good bacteria dies and bad bacteria happens to destroy our protein mechanisms and biochemical activities and all. So they are testing gut microbiota studies, and they are doing metagenomics and all that.
So these are all excellent topics. Thank you for pointing these things. These are relevant in today's world, and we can model it, all of those ones. Again, don't think about how you will write equation at this point. It will come naturally, right? And also I want to stress on this fact that the way we are doing it is not a traditional way of training us. It is a very different way of training. It is about doing it yourself rather than getting a lecture from me on everything. I will give a lecture slowly and slowly in the, today and tomorrow and day after tomorrow and so on and so forth. But that would be, you have to find a lot of information yourself also. So you have to be proactive on that. So having said that, today we will start with, so what did we cover yesterday, by the way? Tipping points. So what was the title of the lectures that we covered yesterday? What was the first topic we covered? Dr. Dharmendra Tripathi covered what? Biofluid. And why did he talk about those things? What was the applications for those?
Some basic concept of fluid dynamics.
No, but why is it relevant? Where does it use? What areas do you use it? In health sciences. In health sciences. Drug delivery. Where? Drug delivery in the body, right? You know, you eat painkillers, for example, or you eat some medicine. It goes to the target tissue. How does it go? You have to build the mechanics of the models so that drug travels in the right directions and doesn't get wastage a lot. It always gets wastage, but most of the drugs get targeted in the tissue, affected tissue, and that's one of the very relevant issue of that. What is the other applications of that? What is the other application of this fluid dynamics or concepts of fluid dynamics in health applications?
There are many kinds of biochemical reactions also going on in the body at many parts, and in which fluid is important, its flow. And like rightly you said, drug delivery, composition of drug.
Okay. You mean to say the not only it is going to the target, but how it is interacting with different molecules of the body.
Yeah, and there are many biochemical reactions in different parts of the body is happening.
So where is the fluid dynamics coming into picture?
Yeah, it is very important in that way.
Okay.
It has chemicals.
So the fluid, what do you mean by fluid?
Fluid and gas and liquid.
Right, but you don't have a gas, so you have gas in the body, for example. You have a blood, right? You have a blood. And blood is a medium in which the drug is flowing, or your immune cells are flowing. Right? That's how you get, if you get hurt somewhere, all the immune cells jumps to that region. If you understand, not everyone's behave in a different way in terms of immune response. Right? So the fluid dynamics concept also helped in understanding how to improve one's immune response also. Right? So there are biomedical application, a lot of them. It is also used in medical devices. These things, these concepts. So there are lots of application in health sciences. I want to point out some applications. Now, second topic that we did yesterday. So this was first lecture by Dr. Bhumin Tripathi, right? And then second lecture we talked about what? Professional skills. We talked about professional skills, right? Third topic, model thinking, right? Which was different than, it is very easy for me to give a lecture on models and analysis. It is very easy, but I don't want to give you the same thing that you will learn it in the classroom as a classes. I want to give you something that you will never get exposed. Okay, so now what I will do is, I will talk about so this was the yesterday's part, right? And then we talked about group projects also like a little bit. So we will talk about epidemic math models, and in order for me to give you some ideas, let me give you first of all what is my background. So I graduated in scientific computing from IIT Kanpur. Then I did, I went for a research scholar at Cleveland Clinic, is a hospital chain in US, and I did. I was part of a department of infectious disease there. So I was there for some years. I was also part of a global health department in Case Western Reserve University, which is in Cleveland. And here, actually, I was part of a clinic. Note one thing, I was trained as a mathematician. I had a mathematics PhD. And I am going through this path, which is all medical sciences, after my PhD. And it is my choice that I went to take this path. And it's again, the reason I'm telling you, how did I end up having different types of skill set? So when I was part of this Global Health Case Western Reserve University, I was part of a group which was working on lymphatic filariasis. It's a disease called as elephantitis. Have you heard of elephantitis? Elephant-like legs or extremities, in fact. Hands and legs. Some people have very big legs, and basically the reason they have this big, because lymphatic system circulates the lymphatic fluids in the body, and in that these patients, these lymphatic glands get spoiled by some insects, some worms, and therefore they cannot able to pump the liquid lymphatic fluids in the body. And so lymphatic fluids get collected in the extremities, and that's why you see big legs and big hands. These people have a lot of problems in doing day-to-day activities. Okay, it's very common. There is in Orissa, in Jharkhand, there are lots of cases of lymphatic filariasis. I worked with Indian Council of Medical Research on this project for some years. But I was working in Papua New Guinea, which is an island country in north of Australia. You know Australia? There's an island country, it's called Papua New Guinea. And why I was there? Because it has the highest prevalent of lymphatic filariasis during that time. And so we were part of a clinical trial. We were distributing medicines free of cost to them. And the hope is that you have the medicine so that if the disease, people get the disease, the medicine is already there in the body to react immediately. And these medicines do not have very slight side effects, so it is very easy to take it. So I was part of this global health, therefore I was part of a clinical trial then. Then I was a faculty at Arizona State University as I was telling, School of Human Evolution and Social Change. This was very interdisciplinary department where it was anthropology, biostatistics, epidemiology, mathematicians, all. There were selected people in one department. So it was very interdisciplinary department. Then I was also at Illinois State University and I'm still affiliated with them. And I'm now a senior fellow at Kalam Institute of Health Technology, which is in Visakhapatnam. It's, as I was talking about, Medical Devices Park, National Park. It's in It's one of the largest park that I've seen in India on medical devices. Now I also have a company called Kalam Experts. This is in India. And I'm also working some projects in industry, and I have a company in US called America IT, which is, I forgot to list it here. So here I am, this is my hometown, and this is the institute where I work in Illinois. Illinois is the state where Chicago, this is Chicago, and that's where it is, and I live here in Arizona. The city name is Phoenix. So now you know where I am, where I'm living right now. And currently I'm working with the two groups. These are modeling group and disease, very famous groups for basic computational epidemiology. If you go on this line, you will see number of projects. We are helping Center for Disease Control in US. So US government is tracking many diseases, influenza, it is tracking vector-borne disease, it is tracking COVID. Naturally, they do it all the time, 24/7. And so we are doing it through that. Another group that I'm part of is called VIPRA, Viral Infection Propagation through Air Travel. So how the infection propagates from one country to another through air travel. Note one thing, the infection cannot get traveled in an aircraft. Why? The infection cannot get transmitted in the aircraft. Why is that so? And still, when COVID happens, the infection was going from one country to another. Still, why we cannot have infection in the aircraft? Any ideas? Because it has a very good air circulation system, very, very efficient system. It circulates, cleans the air very fast. When the flight is on the air, it's do the circulation, and that's why you don't feel suffocation in the long flight, even you going intercontinental flights. You don't feel suffocation. It has a very good air circulation system. So in spite of that, there was infection, and during COVID, the whole world got infected. And primarily because of air travel. Because infection happens during boarding and deplaning. When you come out of the aircraft, when you enter the plane, that is the time most of the infection occurs. Transfer of the infection occurs. Okay. So we are looking into this, and we are working with many airports authorities and airlines to talk about this. So this is, go about and check some of these websites. You will see the type of work that we are doing here. Now this is one of the work. It is published in Royal Society Proceedings. It's on high school dropouts. He was my graduate student. He's now faculty in one of US university. He's very well-known professor in University of Texas, Arlington, in mathematical biology. He's very well known. His name is Christopher Cripps. and he's also a professor in University of Texas. And this work, even BBC, when we published this work, the BBC had a special show on this work. And this was basically what it was looking for is why do certain schools have a very high dropout rate and certain other schools do not have? What is the reason? And so we did this analysis of, now in infectious disease curriculum, a coursework, why I'm talking about high school dropout rates. Why is the high school dropout rates relevant here? Because dropout, it's a dynamical modeling, of course it is one thing, but there is other aspects also. Dropout occurs with social influence. People group in itself and influence each other to drop out. The children we are talking about. And we measure that thing, how much dropout is happening due to social influences. Okay, so, and we did a survey to measure that thing, by the way, in this study. So it is very relevant. Infectious process is happening, but infectious not in terms of a pathogen infection, but a dropout infection is there. Okay. SIAM, Society of Industrial and Applied Mathematics, have an article on this. It's called Silent Outbreak of High School Dropouts. It's published in a SIAM news article. So anyway, and it has a lot of complex bifurcation. Once we designed it, basically it is, there are students who are passing, students who are vulnerable to getting failed, students who are failing, and these drop out students, already drop out. Do you understand what is dropout? Yes. Dropout are people who have left the studies, basically. It happens all the time, right? So that's what we were capturing, and we captured their parents, we captured their social environment in which the dropout students are impacting other students to drop out. So it is not the influence always happens in a positive way. Sometimes encouragement happens in a positive way, but also negative things happen. And dropout is a negative influence that we are talking about. Okay. I have another work that is published a long time back. It's in PLOS 1, and it's on mass shootings. You know, in US it is very common, mass shooting is very common. You know what is mass shooting? Like someone with a gun shoots in a mass and kills or injures many people. So it is a big problem in the developed world. So we studied using models to understand this mass shooting phenomenon. Why is this happening? So this work actually still very highly cited work, one of our very highly cited work. And there is a show by, do you know him? Morgan Freeman, right? Very famous actor. And he does this show called as Through the Wormhole. Through the Wormhole is the name of the show. And so they have one episode of, so they bring different problems from the life. And one of the problem was mass shooting in his show. It's a scientific show basically, Through the Wormhole. Very old show basically. And they have interviewed us. We have one episode through the Wormhole only on mass shooting where our research is being used. Okay. And why is this important for us here? Why is it important for us here? Because what we were looking here was how is this one incident of mass shooting influencing the next occurrence of the mass shooting? Because what happens is, just like when any incident like this happens, it is all over the news, everywhere. So there are good people who are very, good people who are making now more stricter policy on gun control. But there are people who are vulnerable or who are mentally unstable. They are getting more influenced by doing these type of events. So it has these science aware, these news are making people aware of the news, but at the same time it is a disadvantage that the people who are vulnerable and can do such type of incidents are getting more influenced by that event. Does that make sense? So that is what we were capturing here. What is the likelihood that the next event, by the occurrence of this one event, will occur? So we captured that intensity of the event. Okay. And so what we did is, now one thing more, these events, the big problem here is they are not, you can't find the data. There is no data for that. So what we did was we looked into last 10 years of newspapers in the library, and we used some news agencies data also to make and extracted all the details of the mass shooting that has occurred in the last in which 4 or above number of people have died. Okay, and then we basically made the data from scratch, and then we use the models to estimate the parameters. So exactly the topic that, so why I'm exposing these examples? Because it helps you think about your own research project that we are talking about. Now, infectious disease, let me come back to the infectious disease, has many years of, is there any disease occurring right now in India? Is any outbreak, epidemic that is occurring in India? Anyone knows about it? You have not heard in the news, right?
Recently in Lahore, Pakistan,
Yeah.
there's 3
Pakistan.
influenza.
Okay. Say it again. Some flu.
Yes, some flu is there.
Monkey flu. Is that what you say? What is it? Monkey? Okay. Well, there are thousands of outbreaks that are going on right now. Thousands, if not less. Not every outbreak you come to know in the news, or the only news outbreak you come to know are the big ones. But there are always happening the local outbreaks all the time. There could be rotavirus outbreak. Rotavirus is the babies getting diarrhea. It happens all the time in local villages at a very small scale, right? When you work for a public health department, then you get to know these things. But otherwise, in the news, you only get a national level news, right? Or something. So there are lots of outbreaks that happens all the time, and that's the reason you have public health department in every small regions. There is a regional, then there is a state, right? So state has a health department, and then region has other health units of the health department, and they track all these things. So this is an example of some of the major outbreak in the world. You may not have heard about. So have you heard of Spanish flu? How many of you have heard of Spanish flu? Black Death was different. Black Death was not in this century. This is only starting from in the 20th century that I am talking about. So it is 1900 that we are capturing in this picture. But there were other. So Spanish flu, there is a hand there. So what have you heard about Spanish flu?
Just the name, nothing else.
The research paper.
Research paper. So why is it a big thing about the Spanish flu? How many people died? Did you know? Any outbreak? It was all over the world, maximum part of the world. And how many people died? It's the largest outbreak known to the humans that is documented. How many people died? 50 million. Sorry. Okay, I should have typed that. Okay, so there are 50 million people. A lot of people died. That's a lot of people. 50 million is a lot of a big number. What is the how many millions of people are in Delhi? The Delhi metropolitan area, let's say. If you take a metropolitan, it will be around a little less than 18 million or something, 20 million. But if you talk about Delhi only, it will be like 9, something like that, so around that. So roughly around 18, 19 million people in big metropolitan area. So think about 50 million people. So many people, right? So Spanish flu is one of the major ones. West Nile virus, Ebola virus. Have you heard of Ebola outbreak? Recently Ebola outbreak. HIV. Everyone knows HIV, right? Is it still an outbreak?
It's ongoing.
Okay. So this is an ongoing. But there are so many other viruses that you have not even heard of, right? So outbreaks is happening all the times and maybe simultaneously occurring. Measles outbreak is going on in the US. So they are stopping people from going to the Disneyland, for example, because children are getting measles infections, right? So I'm just giving some examples. Coronavirus, there was another coronavirus. COVID was coronavirus. That occurred in 2012, 2013. You guys don't know about it. Not many people died, but it was localized outbreak. It was not global. COVID became a global. When it is a global outbreak, what do you call? What is the name you give? Pandemic. Pandemic. When it is spread globally, it is called pandemic. Right? Epidemic is a small scale. The terms are very important also. Okay, this is now from a modeling perspective, it has huge history. What we relate to the modeling goes back to John Grant, which is in 1620, the first model that we talk about. What do you think? What is the guesswork that you can make? What is the model that he did, John Grant? Any idea? What is the model means for him at that time? There was no dynamical system differential equations that he was writing. Right? At that time the model means, so basically at that time they used to publish all the deaths without telling what is the reason for that. What he did was he categorized the deaths based on types, from water due to drinking water, due to poisoning. He categorized the different tables, and that was the model. So the model was tabulated in a certain categories. What does model means? That it is simplifying to me things that is very complex in reality. So, it's not always when many equation you write that is the best model. The model meaning is different. Writing something that makes people understand things in a simpler way, that is a model. Okay. From there's lots of many types of, for example, Bernoulli's work was on smallpox. Smallpox was also, there's one disease that is eliminated completely from the world, which is? Say it again? Human disease. Which human disease has been eradicated from the world? I'm sorry? I can't hear you guys. Louder. It's a smallpox. That's the only disease that has been wiped out. It's there, but it is there in a lab, in a couple of labs, in Russia and in US. Okay, so there is, it's not, the pathogen is not circulating in the population. It has been eradicated. And only smallpox is the only human disease that has been eradicated. There is no other disease so far that we are aware of. There is many animal disease that has been eradicated, but human diseases are only smallpox. Now there are different types of work. These are all important people who have added some features to the modeling work. For example, rate of contact by Hammer. He described rate of contacts. He described how to write the rate, how to estimate rate of contact. in a very simple way. But that is now what we use it so casually. But these are the people who are pioneer in identifying some features of the models that we use today. Sir Ronald Ross, how many of you have heard of that? He was the first one to model diseases through vectors. What is vectors? Through insects, for example. In this case, insect. He was the first one. In 1908, he got a Nobel Prize for describing the life cycle of the malaria. Life cycle of the malaria means the pathogen in the malaria goes and spends some time in mosquito and sometimes in mammalian host. So there is a cycle there, right? Half life in the vector and half life, not half, but some life in the human host. And that is what he described. Before that, nobody knew why we are getting malaria. They think it is through the infection from coughing someone, right? That's what the people used to think. But now we know mosquito can infect us also. There is other disease, Kala Jowar. Have you heard of that disease? Who is from Bihar? Anyone from Bihar?
Yes.
So have you not heard of Kala?
Yes, sir.
Right. So what is that?
It is a type of illness of mind.
Okay.
Basically, it is a high fever.
High fever, right? High fever, and it could cause mental issues as well. But how is it transmitted?
From any insect or any type of.
Not any insect, especially one insect, which is sandflies. It is called sandflies. And in Bihar, in Odisha, in Jharkhand, it is very high incidence in certain. So, and I worked with there in Bihar at Rajendra Memorial Institute of Medical Sciences on this thing for a state health department. So, have a seat, have a seat. So, so leishmaniasis, it is called Kala Jowar in India. In English, it's called leishmaniasis. And so, Ronald Ross was not a mathematician. Was not a mathematician. He was a doctor, physician. All the mathematical things that we use, which are practical today, most of them are somehow discovered by non-mathematicians. Practical aspect. So the models that we use for malarias are his two students, Kermack and McKendrick. You know about Kermack and McKendrick? Right. So, well, SAR was already established before that. It was not. So it was a reproduction number that was Kermack and McKendrick. This was in 1927. These two were the students of Sir Ronald Ross, and they basically, expanded the theory of infectious disease modeling that we do today, basically. And this is around 1920. This is a very pioneer work. It's the tipping point that we were talking about was discovered during that time. Nobody knew for a long period of time until very late. Okay, so this is early 20th century, 1930s around end. Now this is latest. This is the latest till 2020. Now I want to say this is the first simple model, John Grant, that I am talking about. But also Ronald Ross, 1902, and then there is a gap because I don't want to show everything. Very important, 2002, there was a foot-mouth disease outbreak. Have you heard about that outbreak? It happened in UK. This happens in kettles. So there was farmlands, and this infection was spreading in kettles. Kettles, you know kettles, right? And because this infection was spreading, they have to kill a lot of animals. So there was a huge loss to the farmers. So it became a big issue when the UK were, very dominant at that time in 2002. So there was a lot of modeling work that came forward during that time. It's actually new pioneer work that started around that time. Also there was 9/11. Do you know 9/11, right? 9/11 was the Twin Tower was attacked in US, right? During that time there was multiple things happened. There was anthrax attack. Anthrax was also an infection that was sent to people in an envelope, and when they opened the envelope they got infected, right? Although it didn't become a big outbreak because it was controlled. So at that time, this is a time when modeling work is start to pick up. Why? Because US government was already struggling with 9/11 because they were afraid. Then there was disease attack, and then they were seeing foot-mouth outbreak in UK. So they were very afraid about all these things. So they established a task force. Why I'm talking about US? Because this led to the lot of publications, lot of work that we use as a reference today, modeling work. So 2005 there was a task force that was established by the US government, which has anthropologists, which has biostatisticians, which has mathematicians, which has physicists. It was many different fields in one task force. And their goal was to handle the infection or the risk associated with the infections. So they started doing novel innovation and research work. Task force was pumping a lot of money. So money lead to more research and innovations also. So, and therefore, the Spanish flu that we discussed just now, nobody knows about Spanish flu until 2010, when this task force starts to publish a lot of papers. Then we realize about Spanish flu. In fact, we didn't know what is the virus that is infecting Spanish flu. When Spanish flu was spreading in 1918, nobody knew that it is a virus. They used to think it is a bacteria. Nobody knew at that time. Until 1950s that they realized there is a virus also. And that which virus was the influential in Spanish flu was identified during 2005 and 2010. Very late. We came to know about Spanish flu in 2010, literally. And there was lots of modeling work during this time. And so then it started, see, this is the what is biosurveillance and preparedness task force. This is what 9/11 anthrax. There was actually COVID-1 during this time also, coronavirus 1. What we saw was very late, different types of coronavirus. Okay, studied 1918 flu. So this is the time, 2005 to 10, there's number of papers on modeling work, mathematical epidemic modeling. Then came influenza forecasting. So influenza is something that impacts us everywhere. We all get cold symptoms. You know, we don't care in India, but in Western world they have models to forecast when there will be infections, how many cases, and there are vaccines also for that. So that is started in 2013 onwards, and then there is a Ebola and Zika virus. I don't know if you guys know these things. Ebola and Zika were big news during that time, in 2015 onwards, around 15. So there was this thing. And then there are ensemble model. Ensemble, you know what an ensemble means? It's a collection of many models, not one model for outbreak. The researchers are making many group of models, and because one model was not doing a good job, so they needed a different types of assumptions, different models, and take the results as an aggregated result, and they called it as an ensemble model forecast. CDC nowadays practice this thing. In fact, all of the organizations which are health institutions doesn't take one model, but take a collection of models. Okay, so that was the history that gives us a beautiful pictures about how things have evolved in the modeling world. Weather forecasting, we do it very good. Why can't we do infectious disease modeling forecasting? Very good. Weather forecasting, do you get the weather, right? You check the weather before going to the certain place, and it can tell you precisely for 7 days forecast. but you can't get it for, they don't have any app for infectious disease. Why not? There's a problem. What are the problems? Data is one thing. And what is the problem with the data?
Like, maybe manipulated with the people who are reporting it.
Okay, people are reporting it, right? Wherever people, how is the data for weather is collected? By machines. There are machines at different places. They talk about wind, they talk about temperature. There's no humans involved there, right? Wherever humans starts to come into pictures, things become messy. So the data is a big issue. He's absolutely correct. We don't have a consistent collection of data from every angle. Or if it is collected, it has a lot of issues with the data. It is polluted data, polluted in some sense, right? Also issue is human behaviors. Human behavior is not consistent. It changes. Why behavior is important in disease? Because think about that. If everyone during COVID times have stayed back home, did COVID have a spread? No. Since we changed our behavior, either we were taking more risky behavior based on our knowledge of diseases, we were doing sometimes assisting in spreading of infection. So human behaviors are not consistent and everyone is taking its action individually. So there's a lot of complexities involved in human behaviors, right? That's the reason diseases are never able to be tracked as you can track weather, right? Other things, yesterday someone was talking about one more important factor. What is that important factor in diseases? Evolution, right? Strains, different infection strain evolve. We don't know how the strain will multiply and become more infectious or less infectious or which region it will go. Does that make sense? So the evolution of the strains we don't understand. There are not established models for that. Okay, so that's the distinction that makes it very hard for us to really do the same thing what we can do with epidemic models. In diseases, what do we answer? What type of work we answer? We answer things about vaccine, whether which age group we should give vaccine, whether there will be a delay of 10 days in giving vaccine, how much impact it will do to the society, so on and so forth related to vaccine. Diseases on therapeutics mean drugs. These are the questions we ask. What type of drugs we should promote by government, what we should not, because government doesn't have infinite amount of money. It's always resources are limited. So where should you focus? If you have 100 rupees, where should you focus more? Which disease, which areas? That is decided by models, right? Diagnostic is diagnosing disease, right? Or so government provides a lot of these things. So they have to make models to decide on where to give what type of diagnostic. Market size, how big is the market? Policies, whether young generations could get vaccine, whether older generation could get vaccine. What is the cutoff? What is the cutoff of the age group in which you should give vaccine? You can't give vaccine to everyone because there's no infinite amount of money. I wish, but we don't, right? So these are the issues that we answer through the models. Okay. What's the time? 10:15. What time is the break? Do we have an idea?
11:15.
Say again?
11:15.
11:15. Okay, so we have some time. So microparasite and macroparasites. Micro is viruses, protozoa, bacteria. Macroparasites are helminths. What is the difference? Why are we dividing into these two things? Because the models of microparasite and models of macroparasite are very different. Different models we do, different types of equations we write. This is the difference. Average lifespan is shorter in microparasites. They last for a few days only. Macroparasites could be few years. Example is worms. You know, guinea worms. The Phylactis is also worm. These are worms. Worms, you know worms, right? These are worms. Whereas these are viruses, protozoa, bacteria formed as the microparasite. Other issues is, this is one of the important issues, this one. Parasite. These multiply very small amount in microparasites in a small amount in the human host. Whereas macroparasites, some hosts have very few of the infections, some other hosts have a lot of parasites. It's a lot of variation in the parasite count inside the body of an infected person. So, macroparasites has lot of variation of number of count of the pathogen inside the body. Okay, why is this important? Because we will see it in a few minutes. Also, one important thing is, how is this thing spread? How is virus spread? By cough, cold, right? Direct contact. Direct. But helminths or macroparasite cannot be spread directly. They need another one, such as they need snails. For example, snail is a animals and they live in water, so they can also transfer certain health worms. So they need another insects to transfer. So basically, these pathogens have some life in the human host and some life in the non-human host. Okay, so this is very, very important, and because of that, the models are different in microparasite and macroparasite. Do you understand these compartments? Anyone have seen this? So in microparasite, in virus diseases or bacteria disease, we model it as the population. We are modeling it in this group. We have susceptible people, we have infected people. S stands for susceptible, I stands for infected. So that's a susceptible and infected. This is a microparasite, viruses and bacteria. But in macroparasites, you don't talk about susceptible and infected. You talk about number of pathogens in the human host. The quantity of infection that you are counting. You are not counting number of people who are infected. You are counting amount of infection in the person. Average amount of infection in the person. You see the distinction? That's the distinction. We do have to model it very differently. Okay. But there are also model of co-infection where you have both microparasite and macroparasite infected in the same human host. For example, lymphatic filariasis patients are infected with the flu. So then there is the co-infection cases. Okay. Okay, so how do we model? Model is bookkeeping exercise. I think many of these things you might know, but bear with me because this is important for me to continue in a fluid way. So it's a bookkeeping exercise. So basically it's counting at different time points how many these geese are there, for example, or this is a type of a cat, a wild cat. So you count the basically, and this is a bookkeeping exercise. Bookkeeping is you are noting down the number at different time points, right? So the way you model, if you want to know how many, what is the population in next time step, you take number of people in the current time step, you add the number of births, subtract number of deaths, immigration, how many moved to this region, how many moved out of this regions, and that's the total count of today's, right, based on the last time step, right? That's how we count. That's a bookkeeping exercise. It's like noting down level of the water or amount of the water in the container where you have a tap, two taps which is feeding inside and two taps leaking out, and you are counting how amount of water in the bucket. Okay, that's the same phenomena. And in ecology you do it in sustainability, you do it in, you name the field and you do the same thing everywhere. Nothing different. And this is the model. And the model is about how do you capture birth, how do you capture death, how do you capture these four terms. Now this could be captured in a different functional form, different types of functions. So diseases are modeled using compartment, simple compartment, susceptible, infectious, recovered, and there is this term makes diseases as called as infectious process. Infectious process. Same way as influential process that I was showing an example before, like dropouts, like mass killings, right? This things makes it different than a chronic diseases like oncology. Cancer is a chronic disease. Diabetes is a chronic disease. It's not infectious disease. Okay? So this is the term what makes it an infectious process. And it is the interaction. I comes in contact with S and that result in people moving from susceptible box to infected box. Okay. And again, it's a bookkeeping exercise. How many in the current time, how many left, and how many are there in each of the boxes. So, the transmission is this process of this going from S to I. And it depends on multiple factors. It depends on host population size, it depends on contact rates, it depends on pathogen infectivity. Contact rate. What does contact rate mean? What does contact rate mean? Okay. Do you think it is everywhere the same? What should it depend on? Population. So it depends on the density of the population, right? Okay. So the contact rate, and it is not static, it depends on behavior of the population also, cultural issues. It can depend on many, many different factors. Why I'm saying there are certain religious aspects that covers your whole body, less likely to have this. You can't define the context the same way as we are defining contacts here, for example. Right? So these people spend the whole dissertation in measuring contact rate. I've seen at least 2 dissertations on that. And it is a lot of, and think about that. If we are